Well, hello everybody, and welcome to the 58th Chestnut Chat. We are very honored and happy to have Dr. Duane McKenna and Michael Alexander Charles, who is a student in Dr. McKenna's lab. Dr. McKenna is the William Hill Professor of Biology at the University of Memphis, and the founding director of the Center for Biodiversity Research and the director of the Agriculture and Food Technologies Research Center of the FedEx Institute, which is associated with the University of Memphis, I believe. And these researchers are going to tell us a lot more about a very important insect to the chestnut tree. And I know some of our attendees are growers of chestnuts for nut production, and I know these folks have an interest in the chestnut weevil, and we want to hear more about how the weevil seems to be making a comeback. It was thought to be gone when the chestnut itself, the American chestnut, largely disappeared. So we're going to hear more about this, and there's a couple other things I just want to touch on before we get started. One is that I'd like you please to use the Q&A rather than the chat if you have a question. The chat tends to get a little cumbersome when people could put questions in there, so the Q&A is better, and I will be monitoring the Q&A, and there may be some questions that I can answer or that aren't really relevant. But we're gonna hear from Dr. McKenna first. I guess you prefer to be called Duane, right? But anyway, so we're going to hear from Duane first, and then Michael, who's a fifth-year PhD student in Dr. McKenna's lab. We're going to hear from these guys first, and then we'll have time for questions. This is going to be the last chat for a little while because most of the key researchers in the TACF are really going to be tied up with field work this summer. There's a lot of genotyping that's going to be done. The big focus on recurrent genetic genotype selection, the RGS approach, is going to occupy a lot of key people's time. So there will be a pause for a little while, but it'll be announced when the chats start up again. And in the meantime, there's a wonderful archive there. You can go on to, if you Google Chestnut Chat, you'll find it, and I'll post the URL in the chat also, where the archives are housed, and you can go and listen to previous Chestnut Chats, watch them on YouTube, and it's a tremendous resource. So I encourage people to check that out. I'm going to listen again to a number of them because there's all kinds of stuff. So let's get started. Duane, I'm going to turn it over to you, and thanks again for being here. Looking forward to hearing what you have to say.
All right. Thank you, Mike. I'll go ahead and share my screen. All right, so let me just mention as well that I'm going to talk for 15 to 20 minutes. My focus will actually be on higher level work with the order Coleoptera with beetles. That's my specialty, and I'll introduce that work to you. Michael, who you'll spend the bulk of your time with today, will be sharing on his work with the greater chestnut weevil. So anyway, maybe tolerate a higher level beetle kind of conversation, talking more generally about the evolution of specialized plant feeding. And so the work in my lab ranges from more traditional insect systematics to genomics, with a real focus on evolution and diversity. And in particular, a lot of that work is trying to understand the interplay between ecological opportunity and evolutionary innovation at the insect-plant interface. So theoretical work, but really focused on understanding how ecology scales to produce the sort of really remarkable diversity that we see in the order Coleoptera. So what I'm going to share with you in the short time today that I've got is sort of a background, get everybody on the same page, introducing the order, talking about what we understand and what we don't about the relationships among major groups of beetles, and then with a particular focus on the evolution of specialized plant feeding. So most, but maybe not those who haven't worked with beetles, know that they characteristically have the front or forewings hardened to form elytra. And that's perhaps their most sort of easily identifiable character. Because of this, their flight is powered primarily by the hindwings. They're dorsoventrally flattened as well, and so very well suited to wedge pushing and working and feeding in enclosed spaces. Their mouth parts are adapted for chewing, and as such are different from at least some other insect orders. So I study beetles. I think for a lot of people, it's pretty natural that they'd be drawn to a group like this, but I do find myself often asked why, and for that matter, I think there are a lot of other really amazing groups of insects, not alone other organisms. So why do I study them? So I've always been deeply interested in the evolution of taxonomic diversity. And we all know there are lots of beetles, lots of species, lots of higher level groups, a deep evolutionary history, a lot of undescribed, uncharacterized diversity. They are arguably the most species-rich lineage or order of insects. Their morphological diversity is similarly remarkable, with really striking variation in the body shape, size, and other modifications, measured in a number of different ways. You know, if you think about that as translated into their physiology, it's kind of fantastic and remarkable, right? I mean, you've got the forces of cohesion and adhesion occurring and happening at various and different scales in the different and varied bodied parts of beetles, you know, from the smallest of them to the largest of them. And those are some pretty remarkable and different forces. So here you can see the smallest known adult beetle. That's Scytocella musawacensis, a tiliid. And these are small beetles associated with fungi. An adult is pictured in that blue circle in between the mandibles of that huge Titanus longhorn beetle, a prionine cerambycid, which is by body length the longest known beetle. And so, you know, remarkable variation in scale, similar body parts, similar internal anatomy, all of it functioning at these scales in really fantastic ways. Functional and ecological diversity is similarly varied from plant feeding to predation to fungal feeding, inquiline scavengers, parasites. Now, of course, characteristically, beetles are occupying terrestrial habitats, but they also occur in nearshore marine environments and certainly are diverse in fresh water. They have distributions that include the Arctic and subarctic or subantarctic, and of course, all terrestrial habitats. It's often overlooked that they're exceedingly important pollinators. In fact, they were among the earliest pollinators, and I'll return to that a little bit later. Most of my work, as I mentioned, though, is focused on understanding the evolution of specialized plant feeding. And more than half of all living beetle species are associated with plants. So I'm going to start by framing the phylogeny, giving you an understanding of relationships. And, you know, it wasn't all that recently that we really didn't have a good understanding of how the major groups of beetles were interrelated. And so until about 2019, the relationships were pretty significantly contested, not a lot of consensus. It's true that we knew what most of the major groups were. They had been established pretty well by morphology and existing DNA data, but the composition of the higher level groups, or I should say, while the composition of the higher level groups had been established, their interrelationships had not been. And that was largely because until that point, we were dealing with small DNA sequence data sets, lots of taxa. You know, all of us were wonderful stamp collectors, if you will, but we had not yet been able to generate the scale of data needed to produce a statistically robust phylogeny for the group. So most of what I'm going to be sharing with you today is the result of a paper published in 2019 that incorporated a lot of different kinds of information about beetle phylogeny and the evolution of beetle genomes. While the paper is dated 2019, we've dug much deeper into both the phylogeny of beetles and definitely into beetle genomes, genome evolution, and the evolution of specialized plant feeding since then. But a lot of what I'm sharing with you today is kind of a broad overview, which this paper does a nice job of presenting. So we analyzed two large molecular data sets to establish the relationships. One was nearly 5,000 genes, which, if you think about it, is pretty cool. It's about 1/4 of genes in a given insect genome, or beetle in this case. And most genes aren't well suited for phylogeny, and so this is about as many as can be reasonably used. These represented about half of the extant families, and certainly all the major ones. There was a corresponding data set, which was fewer genes, 89 genes, from across 510 species representing 140 families. So a broader taxon sample providing more resolution of relationships. So a pretty substantial molecular data set that allows us to explore the phylogeny in ways that we had not been able before. It also provided a framework for establishing the timing and patterns of beetle diversification and evaluating different hypotheses about beetle evolutionary history. The data were big as well. They included transcriptomes and representative genomes from across the group, which also allowed us to explore the evolution of specialized plant feeding and the genomic architecture, the genes, if you will, underlying specialized plant feeding. So those trees that resulted were largely congruent, the one from the large number of genes and the one from fewer genes but more beetles. And we resolved more or less all of the higher level relationships, except in one case for the interrelationships among the suborders. And that's not something that would be, or that is incredibly important for understanding beetle biology as much as it is important to those of us that study beetles and really want to have all the questions answered. And so those deepest splits are a bit of a challenge because the branches are very short. And so they happened a long time ago. The signal there is obscured to some degree by reversals, which is effectively molecular convergence. And so we're using other methods at the moment, including the presence of genomic rearrangements and other things to establish the deepest splits. But at any rate, what we established, which was known to some degree from the fossil record, but was bolstered by these data, is beetles have a Carboniferous origin. More than 300 million years ago, diversification rates were not exceptional, which is consistent with previous studies. And this holds in many ways for a lot of insect groups. It would seem, in fact, that lineage preservation, more than extraordinary diversification rates, has played a role in the accumulation of extant diversity. So Polyphaga is this one lineage of the beetles that includes most described living species. And that lineage, which includes most of the plant feeders as well, underwent episodic radiations from the late Carboniferous, where there was a rate increase, and then 13 different post-Permian increases in descendant lineages. So in other words, there's been these radiations occurring over and over and over again at different times in the history of the lineage, correlated in some cases with biotic events, other ones beyond beetles themselves, relating to flowering plants and other biology, and other events which were geological in origin, it would seem, or environmental, if you will. So at any rate, I'm not going to dig too deep into any of that, but to say that beetle evolutionary history is exceedingly long, and not surprisingly, that history is intimately tied to other Earth scale events over its history. So the lineage itself diversified into 180 of the 188 families that we sampled by 65.5 million years ago. So that was the Chicxulub KT meteorite impact that wiped out the dinosaurs. And at the same time, what we learned by this is that beetle diversity is really old. So literally all the families, with the exception of less than 10, were present more than 65 million years ago. They also survived the Big Five mass extinction events, including the Great Dying at the end of the Permian. And so beetle diversity is both old and persistent in the sense of relatively low extinction rates. Sure, a lot of species were lost by those events, but higher level groupings, it would seem, not so much. Beetle is associated with gymnosperm pollen. So of course, gymnosperms diversified before the angiosperms alongside beetles. And it would seem that beetles that are associated with the cones and other reproductive parts of gymnosperms appeared really early. They appeared by the late Jurassic before bees and butterflies and were therefore the earliest important pollinators of these plants. The prevalence of transitions from generalized diets like detritivory and saprophagy towards specialized diets like phytophagy and mycophagy has occurred. It's important, it's prevalent, it's consistent with lineages that are diverse within the beetles. There was a statistically significant diversification rate increase in 2 plant feeding lineages. And these will be familiar to most of all, if not all of you. The buprestids, see, this is the jewel beetles or metallic wood boring beetles, as some of you know them, and the phytophaga. So the phytophaga is a clade, it's a monophyletic group comprised of the leaf and longhorn beetles, that's superfamily Chrysomyloidea, and Curculionoidea, which are the weevils. So that clade is exceptionally species rich. And like the buprestoids, the buprestidae and schizopodidae comprising the buprestoids, are plant feeding, and in particular specialized plant feeders, including subcortical wood borers and other kinds of habits. The other thing I'll point out here is that these 2 groups that experience these significant rate shifts, increases, are not close relatives. They're separated by more than 250 million years of evolution, and their nearest ancestors have other habits. So they have independently evolved plant feeding, specialized plant feeding. So I'm going to show you the phylogeny of beetles, just a sort of brief, quick sweeping overview. So there's a suborder called the Mixophaga that is sister to a clade comprising suborder Archostomata and suborder Adephaga. The Adephaga are the ground beetles and the whirligig beetles, and again, familiar, charismatic, diverse. There's about 45,000 species of Adephaga. Archostomata had their heyday back in the Permian. They even predate the Permian. And so at that time, they were the predominant beetles. They are the predominant beetles in the fossil record. Today, there's only 50-some species extant. Mixophaga are similarly species poor today. They're infrequent in the fossil record, though we're starting to find more and more of them now that we know what to look for, because their early fossil record is both obscured by these being exceedingly small beetles, and some of them being relatively different from members of the extant fauna. And so at any rate, the Mixophaga and Archostomata are these really interesting groups that persist today, but are relatively species poor. The Adephaga, of course, present worldwide, relatively diverse, mostly predaceous. The remaining groups that you can see here in green are the Polyphaga. And these are perhaps most familiar in part because of their associations with plants. The Skirtoids are sister to the remaining extant Polyphagan Coleoptera. There's another lineage called the Clamboids, the Rhinoripoidea, the Dasyloidea, the metallic wood-boring beetles, the Beroids, the Elateroids. These include fireflies and click beetles. Nosodendroids, they feed in sap flows and are a really peculiar and phylogenetically isolated lineage. The Hysteroids or clown beetles, Hydrophyloidea, the rove beetles, the Staphylinoids and relatives, the scarabs, the Scarabioids and their relatives, Bostracoidea, Claroidea, Coccinelloidea, these are the ladybirds and their relatives. The ship timber beetles, Limexilloidea, are sister to the Tenebrionoids, and in many ways form a very nice clade themselves. Erotiloidea, these are interesting and varied in their biology. Nididuloids, sap beetles. Cucujoidea, which, as you can see here, traditionally had been viewed as forming a single clade, including the nitiduloids and erotiloids, but we then found that they were a paraphyletic grade. They're now recognized as these 3 separate superfamilies: Erotiloidea, Nitiduloidea, and Cucujoidea. And then the Phytophaga, the leaf beetles, longhorn beetles, and weevils, forming this clade of extraordinary extant diversity. All right, so I want to transition then to the work we're doing to understand the evolution of specialized plant feeding. Before I do, though, I'm going to skip back because I meant to highlight the phylogenetic placement of these two really species-rich plant-feeding clades. So we've got the Buprestoidea, which if you look is in sort of an aquamarine blue near the early part of the polyphaga. There are about 16,000 described extant Buprestoids. They are, as you know, mostly wood borers. On the right-hand side of this tree, distant phylogenetically, and as I mentioned, it's actually 250 million years of divergence since they last shared a common ancestor, are the Phytophaga, the leaf, longhorn, and weevil beetles. Now, these aren't the only plant-feeding beetles, but they are, by quite a distance, the most diverse lineages of plant feeders. So many families of beetles then have at least a few species that eat plants, but most species belong to those two clades that I just revisited. Together, they comprise an extraordinary range of plant-feeding habits and behaviors, including chewing, mining, galling, boring, you name it, all kinds of variations on each of those. They feed collectively on living, dying, or stressed, dead or decomposing plants and their parts, and virtually all parts as well. Most species are relatively specialized, feeding on specific plant taxa and parts of those plants at specific stages in the plant life cycle. But collectively, they can do pretty much everything. The genomes of some plant-feeding beetles, we've learned, encode carbohydrate active enzymes that degrade plant cell walls. And when I say we, I mean collectively. This is work that's been done over a very long period of time by a lot of different people. And we're standing on the shoulders of a lot of different people's work in the work that I'll be sharing with you in the coming slides. So these endogenous, that is encoded in the beetle genome, plant cell wall degrading enzymes, include carbohydrate esterases, polysaccharide lyases, and glycoside hydrolases. Together, they enable digestion of cellulose, hemicelluloses, and pectin in plant cell walls, which of course, as you're probably well aware, are the most abundant sources of carbohydrates on Earth. That's a pretty big innovation, a pretty important metabolic capability. In doing so, they liberate sugars, amino acids, and other essential nutrients from that woody plant material. Plant cell wall degrading enzymes are required by some beetles, in fact, for successful herbivory. Other insects, including ones that are probably more familiar as degrading woody plant material, things like termites or maybe even wood-feeding roaches you've heard about, they have similar metabolic capabilities, but they are conveyed significantly by symbionts. So they require those symbionts for their biology and for adequate digestion of those plant tissues. So plant cell wall degrading enzymes were originally obtained by beetles by horizontal gene transfers from bacteria and fungi, it would seem. Gene duplication and functional diversification of these genes after their horizontal transfer into the beetle genome has resulted in multi-gene families with complementary catalytic activities. So collectively then, these repertoires of genes and individual wood-boring or otherwise plant-feeding beetle genomes that allow for pretty robust capability in breaking down plant cell walls. These genes, I've proposed through a number of different papers, are key innovations in the adaptive radiation of plant-feeding beetles. And again, this isn't just stuff that we've done. Others have proposed similar ideas. We've taken it a little further, placing it into phylogenetic context, and started to really deeply evaluate this role by establishing, in fact, this in a temporal and phylogenetic framework, which I'll share with you in a few minutes. At the same time that these ideas were first proposed, though, the phylogenetic distribution of these genes in beetles and their impacts on beetle diversification were virtually unknown. They were just observed to be really fantastic and amazing genes conveying pretty cool biology, but it wasn't clear how many beetles had them and whether they were strictly correlated in a statistically significant way with species-rich phytophagous lineages. So through this paper and the work that led up to it, and again, subsequent work as well, We've documented the numbers, kinds, and phylogenetic distribution, and the timing and first appearance of genes encoding putative plant cell wall-degrading enzymes. We've also documented invertases, another set of glycoside hydrolases, which catalyze the conversion of sucrose. And sucrose, of course, is the primary plant storage carbohydrate, breaking it down into glucose and fructose. So that's an important thing for insects to be able to do because, of course, plants don't want them to. And so they've evolved their plant storage carbohydrates in such a way as to avoid, if you will, specialized plant feeding and exploitation of them. So we asked the question, is the presence of these genes correlated with taxonomic radiations and shifts in host range? What we found was that plant cell wall degrading enzymes and invertases are almost exclusively found in the genomes of the buprestoids and the Phytophaga, the most species-rich and specialized radiations of plant-feeding beetles. They encode expansive and remarkably similar arrays of these genes as well. So it's kind of cool. They've actually converged on having the same or nearly the same sets of these genes in their genomes. Various and different copy numbers, and presumably different functions have evolved via sub and neo-functionalization of these genes, but the genes themselves are largely the same. They've been independently acquired, it would seem, at least originally, before they diversified in the beetle genome from fungi and bacteria. Pretty cool stuff. So convergent evolution, no doubt, separated by more than 250 million years of evolution. So here's a summary phylogeny showing you the major groups of beetles and the distributions of these genes along branches in the phylogeny. And on the right-hand side, you can see that the Phytophaga and the buprestoids have this extraordinary array and copy number variation in these genes. We do see other beetle groups obtaining these genes kind of haphazardly here and there across the phylogeny via horizontal gene transfers from the same kinds of fungal and bacterial donors. These are phylogenies or gene trees, if you will, for each of the glycoside hydrolases and a few other genes as well, but that we found in these beetle genomes. The inferred original donors were, in fact, as I mentioned, bacteria and fungi, including ones known as degraders of lignocellulose and other complex polysaccharides in plant and soil detritus. Now here I've noted that the mode of horizontal gene transfer is unknown. I'm not quite ready to talk all about that, but we're in a really exciting time in the lab where we've sorted a lot of this now out. And so we're working on some papers to be able to report the details of this really cool biology that's behind the evolution of these innovations in beetle genomes over evolutionary time. And I might add, though it's not easy to discern looking at this at such a scale with so much information, what we see with these genes as well is that they are both acquired regularly over their evolutionary history of Coleoptera in these two beetle lineages and lost as well. So there's a sort of a dynamic interplay, if you will, between fungi, bacteria, and these beetles over evolutionary time. So I'm going to sort of bring us to a close in the next few slides here. These genes enabled efficient, symbiont-independent digestion of plant biomass, promoting the evolution of uniquely specialized plant feeding habits. Horizontal gene transfers followed by duplication and functional divergence of genes contributed to the addition, expansion, and enhancement of the beetle metabolic repertoire. The diversification rate increases we saw in the buprestids and the Phytophaga. What's really cool about that is they are coincident in the phylogeny, like along the branch we would expect, where we see these lineages acquire these genes, suggesting that these genes triggered adaptive radiations that resulted in nearly half of all living beetle species today. So the evolutionary origins of beetle diversity has traditionally been attributed to the adaptive radiation of beetles feeding on flowering plants or angiosperms. But we know that lots of angiosperm feeding beetle groups are not more species rich than their relatives with other feeding habits. And this is also true for other phytophagous insects, by the way. Many groups of angiosperm feeding beetles then are not particularly diverse, and many non-angiosperm feeders are diverse. So what we conclude from the work, or have concluded, and this holds with additional work that we have done as well, is that plant cell wall-degrading enzymes and invertases enabled buprestids and the Phytophaga entry into new adaptive zones. They didn't need to obtain and maintain symbionts for plant cell wall degradation, even though they do have them. Pectinases and invertases likely promoted the feeding on cambium and sapwood, fruit, leaves, and seeds of all kinds, and not just on flowering plants, to include gymnosperms and others. Hemicellulases may have promoted feeding on wood. We're sorting a lot of this out, by the way, right now through functional work, cloning these genes into insect cell lines and evaluating their function in the lab. Endogenous plant cell wall-degrading enzymes were likely key to the evolution of specialized plant feeding habits, things like leaf mining and seed and stem boring, which required dealing with plant allelochemicals, nutritional and defensive barriers, and recalcitrant plant biopolymers. And by the way, not even a part of the story today, we're also learning really exciting things about their abilities to engage with lignin as well. So beetle diversity, it would seem, resulted from multiple factors: a low extinction rate and long evolutionary history for sure, co-diversification with angiosperms and in the new habitats that angiosperms created, and importantly, the adaptive radiation of specialized phytophagous beetles in association with all kinds of plants, following independent origins of plant cell wall-degrading enzymes and invertases from bacteria and fungi by horizontal gene transfers. So beetles and plants have had a long evolutionary history, starting way back in the Silurian and terrestrial environments, coming up out of marine environments, if you will, together onto the land. Beetles appearing about 100 million years later, radiating alongside gymnospermous plants first, making their way through mass extinction events, as you can see, way predating the dinosaurs, diversifying further alongside flowering plants. And by the way, this date we could push back probably a little further than it is now. Angiosperms rise to ecological dominance, with beetles having had nearly a 200 million year evolutionary history already. So intimate, deeply co-evolved interactions for sure. A mid-Cretaceous increase in diversity and diversification rate of plant-feeding beetles is pretty well established. There was an asteroid or a meteorite impact, if you will, causing the mass extinction that included dinosaurs, but beetles made their way through. All major lineages, it would seem, that are in the fossil record are extant today at that time, with a few families that appear to have diversified and appeared since then. It was warm after that. There was a period during the Paleocene Eocene when the tropics expanded and beetle diversity grew. But as you can see, that history is deep, co-evolved, and really intimate with plants. So lots of people to thank, whether it's collaborators and postdocs, institutions that have loaned material that we've worked with, and certainly funding agencies, NSF, USDA, the University of Memphis, and others. I also want to acknowledge data sources and colleagues that I collaborate with on a larger scale, including the Beetle Tree of Life community, the One Kite community, the 1K Weevils project and community, and the Insect 5000 Genomes community as well. So lots of great, wonderful consortia that have helped to generate some of the data that supported what I shared today. My friends, Peter Neskrecki and Alex Wild, wonderful, extremely talented photographers, as well as Udo Schmidt, who allowed me to use their images in my presentation. So I'm going to end there. Thank you so much. I'm going to quick use the moment I have here to introduce my PhD student, Michael Charles. And if you'd hold questions for me until at the end, because I really want to ensure we have enough time for you to hear from Michael. His work is exceedingly relevant, I think, to most of your interests. And I certainly find it wonderful and enjoyable. Michael's also nearing the end of his PhD, having some time left, but at any rate, in a position as well where he'll soon be looking for what's next. And so look forward to him being introduced to those of you he has not yet met and sharing his work. So with that, Michael, I'm going to stop sharing and I'm going to turn it over to you.
All right, let me get this going. All right, so my name is Michael Charles. I'm going to talk about my work in general, but mostly focusing on my rediscovery of the greater chestnut weevil. So I'm going to talk about a few things, the characters and biology, as well as the host associations of the genus Curculio, and the molecular phylogenetics that I've been working on, as well as the evolution of the genus. At which point I'll start discussing the history of the greater chestnut weevil, a trip I took in 2022 trying to find it, and then actually finding it a year later. So Curculio are rather distinctive in their morphology. They're very easy to recognize. They have an exceedingly long rostrum. Their mouth parts are unique, apart from other weevils, in the sense that they are triangular and move vertically rather than horizontally when they chew. They have elbowed antennae with 7 funicular segments. Their elytra have 10 striae, and they have a depressed scutellar shield along with a substitutional ovipositor. So I'm going to go over the biology of the genus as a whole. So the biology is rather well known and well studied. So the insect will use their rostrum to more or less drill and excavate a path into the seed, whether it be a chestnut, a pecan, an acorn, a hazel, at which point they will turn around and then use their substitutional ovipositor to lay the egg, oviposit it inside of the seed. The larva will feed on it for 2 weeks to over a month, depending on the species. And we can see in this image of a chestnut the damage that they can cause. The larvae, once they are finished feeding, they will exit the seed as seen in the video on the bottom. And once they do that, they will then burrow into the soil. At which point, depending on the species, they will diapause and stay down there and over for a period of 1 to 4 years. And even within any given species, there's going to be variation in that duration of diapause as well. Once they do pupate, it can last for about 1 to 3 weeks. This image is of a pecan weevil pupa. Once they do that, they will wait for a few weeks to a month for their exoskeleton to finish hardening, at which point they will crawl their way up through the soil and emerge and fly up onto the, either crawl or fly up onto the host tree that that is typically going to be above them. So the genus is widespread, found all over the world except Antarctica and South America, and is on a wide variety of host trees. So in North America, most of them will feed on Fagaceae, so oaks, chestnuts. There's 1 species that will feed on pecans and 2 that will feed on hazels, and then I'm sure you all are familiar with the 2 chestnut weevil species. In Europe, again, they will mostly feed on oaks, and there is 1 that will feed on chestnuts. There is also 1 that will feed on hazels, and there's 1, interestingly, that will feed on both oaks and chestnuts. In Asia, the majority of them will feed on Fagaceae, Silicaceae, and Betulaceae. There's 1 that will feed on hazels as well. There are 2 that will feed on chestnuts. There is 1 that will feed on a legume. And there's also 1 additional species from the literature that's reported to feed on chestnut, but I haven't been able to actually find any corresponding literature to verify that. In Africa and Australia, they will almost exclusively feed on figs. Not as much is known about their biology in detail, but the generalized biology of the genus as a whole still corresponds to theirs as well. I'm going to switch over to talking about my research into the phylogenetics and evolution of the genus. So there have been, to date, 1 phylogeny of the genus. It was constructed back in 2004 using 2 mitochondrial and 2 nuclear genes. The resulting tree had rather low statistical support at most nodes due to failure of PCR. And as a result, they were still able to determine that there are two European species groups, the LFS group and the Glandium group. However, placement of the American species were still unresolved, as they had a very limited taxon sampling of only 21 species. To put that in context, there's estimated to be anywhere from 350 to 500 species in this genus. About a decade and a half later, in 2017, there was a phylogeny on the tribe Curculio, which is the tribe that Curculio is in. It was also constructed using two mitochondrial and two nuclear genes, and it was also constructed using a 28S gene. There were representatives from, there was more representation of the species based on geography. So there were some from Africa, Europe, Asia, and North America as well. So it is more, forgetting my words, excuse me, it is more detailed, it is more inclusive, but it is still limited as it only includes 34. The support at the nodes was better, but still not ideal. This study did conclude and agree with two European species groups, but the American species groups were still rather unresolved due to poor to moderate statistical support. The Afrotropical species were recovered as monophyletic, as well as the Nearctic species. And the Palearctic species, so Europe and Asia, were recovered as non-monophyletic. So my goal in reconstructing this phylogeny is to make it the baseline of what anyone would use when studying this group and their genetics and phylogenomics of it. So they're found, as I said, they're found basically all over the world. There are a few instances of them being found in South America, but they're not known to be native there, as there's no oaks in the areas where they're found in South America. So I'm hoping to produce this phylogeny and a time tree to explore the timing and phylogenetic patterns of its diversification, as well as exploring the distribution of the species groups within the phylogeny. More or less, how are they grouping together? Are they grouping together by region, geographic region? Are they grouping together by host association? Is it a mixture of both? Something else entirely? That is what I'm hoping to find out. I'm also hoping to find out the way that they became this dispersed and found nearly all over the world. So naturally, I'm aiming to include as many taxa as possible. I currently have, I'll get to that in the next slide, actually. We're using anchored hybrid enrichment with a probe set that was designed in Dr. McKenna's lab. It targets 522 loci, nuclear loci, and we can routinely recover approximately 400 of those from well-preserved weevils. Mine have mostly been museum specimens collected at various times and in various conditions. Some of them worse than others, some of them collected within the last few years. On average, I'm recovering nearly 300 from these 300 loci, excuse me, from these museum specimens, which is rather good. Currently have 51 species of Curculio and their near relatives ready for sequencing or already sequenced. I have 23 of the 27 species that are found north of Mexico. I have another 28 from Africa, Australia, Europe, and Asia. And I have the genera Tomola and Archarius to use as outgroups to root the resulting tree. I currently working to increase representation of the taxon sampling from Mexico and Mesoamerica, as well as the Palearctic regions. In particular, I'm currently working on getting a loan from South Korea to increase my taxon sampling by 25 So to bring that up to 76 species. Now I'm going to move on to my work with the greater chestnut weevil. So as I'm sure you all know, we have the American chestnut as its only documented host for the weevil. They will use the nuts inside the burrs and use those to deposit their eggs, in which the larvae will feed. And we have a map of the native range of the American chestnut on the right from the American Chestnut Foundation. So as I'm sure most of you, if not all of you, are aware, chestnut blight was first noticed in 1904 in New York. It will typically enter through cracks in the bark of the tree, at which point it will start to girdle the tree and interfere with its ability to transport water and nutrients as it attacks the cambium. While the trees will mostly die, there are some large surviving Americans that are still able to live with the blight. Most of them will die back, and the root system is interestingly not affected by blight. So the root system will send up new sprouts every so often, at which point this will typically just continue happening as a more or less a vicious circle. So to map the decline of this species, I have solicited specimen data from over 50 museums. Most of them have come from the Smithsonian Institution, as well as the Harvard Museum of Comparative Zoology. I have mapped the accumulated specimen data in ArcGIS and have added the layer for the native range of the American chestnut. So there's a varying degree of usefulness in these data as they're varying levels of quality. I have 310 specimen records. Only 184 of them have dates on them. Some of them will have states. Fewer of them will have detailed locality records going down to the city. And very fewer of them will even have geographic coordinates, given that most of these are rather very old specimens. So we can kind of see in this, we have on the right a illustration from Gravatt in 1949 about the spread of the chestnut blight with its introduction in New York in 1904. We can see back in the late 1800s, we had 3 and in the 80s and 8 collections in the 90s, at which point in the 1900s, there were 56 and 57 in 1910. Once the blight starts to spread, we see fewer and fewer records of this beetle having been collected. We go from 57 in the 1910s to only 39 collected in the 20s to down all the way to 3 in the 1930s and remain in those single digits to today. It would appear that this decline in their abundance would coincide to the decline of their host plant due to the introduction of the chestnut blight. So I mapped these locality data and overlaid them with the native range of the American chestnut, and the vast majority of them, as we would expect, overlap with the accepted native range of the American chestnut, with 5 records in 3 counties that lie outside of this native range. One of them is Wilkinson County in Georgia. Another is a county in South Carolina, and there's one up in New York, where there are records in neighboring counties. There are also a handful of records that only have state level data that include a collection from Illinois, 2 from Indiana, 5 from North Carolina, and 2 from Ohio. The black stars are going to indicate the locality where we have found rediscovered the greater chestnut weevil, where they're currently actively reproducing populations today. So now I'm going to talk about a trip I took to State College in the fall of 2022. You can see in this map for my naturalists, there's a bunch of red pins denoting the presence of an American chestnut tree. I went to 70 of these trees, and each of them were examined for the presence or absence of catkins or burrs to indicate reproductive quality of the tree. They were inspected for cankers to see if there was any visible sign of chestnut blight at them. I also investigated the tree for, obviously, to see if there were any curculio on there. I sweeped the branches as well as shook the branches down because these weevils will play dead typically if they're disturbed. And I also looked for evidence of weevil feeding damage. So are there ovipositional scars in the nuts? Are there exit holes in nuts from previous seasons that are lying on the forest floor? And any fallen burrs were opened and looked at to see these chestnuts inside, to look for this feeding damage. So I went to a few generalized places where the majority of these trees were clustered. I went to Bald Eagle State Forest and several other state parks. I went to some recreation areas, some state game lands, and a couple of state forests. These were all in the area of State College. And then I went to, during the same trip, I drove to Prince Frederick, Maryland, to the American Chestnut Land Trust, where at the time, the last known collection of the greater chestnut weevil occurred. We now know that there was a more recent one in 97 that we learned about after this trip from Georgia, but with the information I had at the time, this was the most recent place the weevil had been collected. So 61 trees were examined, and unfortunately only 6 of them were fruiting. Fallen burrs were opened to examine the chestnuts within them. Most of the chestnuts either appeared underdeveloped or aborted, and there was no evidence of feeding damage or any curculio damage for that matter. So unfortunately, I was unable to find it. So at that time, I was kind of disappointed, thought maybe it's extinct, more likely it's only extinct in this locality. At that point, my next steps were to investigate other locations where there were still large naturally occurring American chestnut trees, such as Southeast Maine, where there were a handful of trees that were rather large, upwards of 30 meters tall, and nearly 100 centimeters in diameter at breast height. After this trip, we learned about a more recent specimen that was collected in Georgia in 1997. And given that this locality I was unable to find any herbarium records or any other kind of records of American chestnut being in this county, I was beginning to look into the possibility that it would also occur on other native species of Castanea as well. So now I'm going to get to in my opinion, the very exciting part was actually finding this weevil. So I talked about the biology of the genus as a whole. For each species, it varies slightly. So I'm going to go over the biology for the greater chestnut weevil in particular. So the females will still excavate that hole through the burr and into the nut, at which point they will turn around and use their substitutional ovipositor to oviposit their eggs inside the nut, which we can see in this video. At which point the eggs will be in the egg and the eggs will be in the nut and will hatch after about 5 to 10 days. Again, the larvae will feed for 20 days to 30 days, and they will devastate the nut, usually to the point where they will no longer germinate and are no longer useful for human consumption or human purposes. They won't grow, and I don't know about you, but I wouldn't want to eat that chestnut. The larvae will exit the seed and burrow into the soil, where they will spend the winter as larvae before generally they will pupate the following July. There are occasions where it is known that the genus as a whole, and as well as this species, will kind of use a bet hedging strategy, and most of them will overwinter for only one winter and pupate that following July. However, there's a smaller portion of them that will continue to overwinter as larvae for a second winter and then pupate in that next summer months. After pupating, they will remain in the soil to allow for their exoskeleton to harden, and they will begin emerging typically around as early as July. At which point they will emerge and either fly or climb the tree up to the catkins and burrs above, where they will mate from around August to October. In which point they will deposit the eggs and then the cycle will go over again. So I actually found these populations of the weevil through iNaturalist and Facebook. The first one I found was on iNaturalist and found in Kennett Square, Pennsylvania, on a Dunstan hybrid chestnut. I went there in November of 23 and collected several larvae, and the two largest of them were sacrificed for DNA purposes, which I will get to a bit later. The site was revisited by collaborators this past September, and we found another female greater chestnut weevil as well. What's interesting is that this specimen was observed in 2022, as you can see stated on the website. And during this time, I was in State College, so I was almost in the right area, just a few hundred miles too far to the west. In February of 2024, something told me to look through Facebook at the American Chestnut Foundation's pages and see if they had any chestnut weevil pictures. Fortunately, they did, and I looked at it and confirmed the identity as a greater chestnut weevil, at which point I contacted the Meadowview Research Farms, and they were able to send me quite a few larvae. Again, at this point, the two largest were sacrificed for DNA. So I visited the Meadowview site in August of last year, where I finally personally collected two of these weevils. I was just very excited because it had been a long time I had been looking for these. In this image, we see that the female is currently excavating that hole through the burr and into the nut, at which point she will turn around and lay the eggs. While I was in Meadowview, there were two other posts on iNaturalist of the greater chestnut weevil, both of them at another orchard that is managed by the American Chestnut Foundation at Mount Cuba in Delaware. So the two I found in Virginia were females, and the two that were found in Pennsylvania were also females. These two that were found in Delaware were both males. These were attracted to a UV light that was set up at a hybrid backcross orchard. So for the DNA and sequencing, to confirm that these were, through genetic analysis, that these were greater chestnut weevils, I sampled from each of the morphological species groups that are found in North America, as described by Chittenden back in 1926. And again, we used anchored hybrid enrichment and recovered approximately 220 loci from this group of taxon that we sampled. We used IQ-Tree to build the tree as well. And so we see let's see if I can get a laser pointer on here. We can see in the tree that the larvae all came out to group in a single clade with the two museum specimens that I had sampled, with full nodal support indicated by this black dot. The relationships between those larvae and the museum specimens have lower support, as these probes are made for more higher level taxon sampling. It was found that the lesser chestnut weevil, Curculio to be the nearest living relative, followed by the pecan weevil. Both of these make sense to me, as obviously the lesser chestnut weevil will also feed on chestnuts, and the pecan weevil, Curculio also has a characteristically longer than average rostrum for this group, I would say. I also sampled the European chestnut weevil, Curculio elephas, to root the tree, and it was recovered as sister to the sampled Nearctic Curculio found in North America. This can possibly suggest that there was a radiation of Curculio into North America, and with them becoming to be associated with economically important tree families such as oaks and hazels. So with that being said, there is still a lot I want to do with this beetle. I want to officially evaluate the suitability of alternate hosts, given that they were found ovipositing on hybrid chestnuts. It's clear that they are not specialist on strictly American chestnut. They can make do with hybrids, and I'm interested to see if they can also use other non-native species of castanea, such as Chinese chestnut or European chestnut, as well as our native chinkapins, whether it be the Ozark chinkapin or the Allegheny. I'm also interested, given that these were all found at managed orchards, I am interested in surveying other orchards to see if they are present there as well. With that being said, there are a lot of people that I need to thank for all the work that I've done thus far. Most of the museum specimens came from the Smithsonian and the Harvard Museum of Comparative Zoology. I received funding from the University of Memphis Biodiversity Center for Biodiversity Research, as well as our department and our college. I need to thank Dr. Chamorro at the Smithsonian for allowing me to borrow specimens and use them for sampling to build the phylogeny that I showed you, as well as Dr. Robert Anderson for kind of guiding me in the beginning steps when I was getting started with this group, as he is a renowned weevil specialist on North American species. I also have all of the collaborators that I have for finding this weevil. They include Matthew Sarver and Katie Bird. Katie is at University of Delaware. Dr. Kate Santos and Leah Johnson at Longwood Gardens. Dr. Gareth Powell, who coincidentally was able to send me both of the museum specimens that I sampled for the greater chestnut weevil, as well as the entire staff at the Meadowview Research Farms for allowing me to come out and use their equipment, their lifts, to go up into these trees and actually find this weevil. For the work I did in Pennsylvania, I need to thank the land managers for allowing me to come and survey on their land. And with that, I will take any questions. Let's see if I can get it.
So Michael, there were a few questions that look like they've been answered already, and everyone should be able to go into the Q&A and see those. I don't know if there's anything that you or Duane want to add to any of those answers that are already in there. I had a question that you kind of already answered in a way, because it sounds like you're working on this, but I was wondering about all the commercial chestnut orchards that there are. And, Michigan has a lot, and they're in the Midwest. And I guess you're gonna try to check those out and see if you find these guys at those places. And I was wondering if you think there's anything different about the Chinese and the hybrid trees that is inhospitable to the greater chestnut weevil in some way. Is there any reason to think that those trees might be producing metabolites or something that it deters these insects, or is that something that's still, not really clear?
I don't have any data to back this up, but I don't think there is, for a number of reasons. One is that I was able to get the genetic makeup of the trees that I found this weevil ovipositing in, and one of them was an F1 hybrid, so roughly 50-50. Another one was an uh, by Gen 2 back cross, I believe. And so it doesn't seem that given that the Meadowview farms have varying large number of trees of varying genetic makeups, whether they even have a couple of pure Americans, as well as pure Chinese and anything and everything in between. They don't seem to be favoring the ones that are back crossed and more American than Chinese. But I don't have any statistical data or otherwise to confirm that they will use any chestnut. That is something I'm hoping to be able to do moving forward.
Yeah. And I see here in the Q&A that Dan Lefevre is pointing out that the Dunstan hybrids are at least they're 90% Chinese, and I've heard maybe they're even more than that. So that since you found one on a Dunstan hybrid, that would certainly argue that there's no big difference in the trees. There are some questions here that look like they were answered, but there's one here. This is also from Dan Lefevre, and this would relate to, I think, potential control of these in orchards. He's wondering whether the greater chestnut weevil can fly and alight in the upper story of the tree, or does it target the trunk, fly towards the trunk, hit the trunk, and then crawl up? And if so, would it be possible to prevent that, the weevils from getting all the way up to where the burrs are with circle traps or trunk applications of a contact insecticide? So I wonder if you know anything about the behavior of these weevils when they're in the process of trying to get to the burrs.
Yeah, absolutely. So they can fly, and I found out I was actually fortunate enough to be able to watch one of them fly um, while I was in Meadowview. Um, I wouldn't say that's their first choice, given they don't appear to be very, let's say they're front heavy, given the exceedingly long rostrum. They can still maneuver and everything, but it's not as refined flight as others, based on my personal observations. I know in the literature they will emerge and then climb towards the tree and can be caught in circle traps and emergence traps as well, all along the tree. So that is a way to control for them. As far as insecticides, um, I would imagine contact ones would be the most effective because you would need to stop the adults when they emerge before before they have an opportunity to oviposit inside the nut. At which point, I don't know enough about the tree itself to state whether or not the insecticide would do any good to the larvae inside the nut at that point.
There's more questions coming along. I've got one more. I don't want to dominate the questions, but back in the day when American chestnut trees were very common, and you pointed out that the weevils, once they get into the nut, they can pretty much wreck the nut so that it won't germinate. But I'm thinking there must have been kind of a balance that would develop where some of the nuts would be viable, and otherwise the tree wouldn't have become so common. And it makes me wonder whether there are predators of these beetles, or other, what would help to maintain a balance in nature so that some of the nuts would still be viable. You wouldn't have, every nut getting eaten up by the weevils. So any thoughts on that?
Hey, Michael, I just would mention, I think what you experienced when you were first looking for nuts on the ground infested with larvae at Longwood. One thing that I picked up from that experience was that still surprisingly few nuts had larvae in them. Is that right?
Yeah.
Apparent greater chestnut weevil larvae versus what were likely to be lessers and what did turn out to be lessers in some of the barcoding work of those larvae. And so I wonder, Michael, if you fully agree with that, and if also you agree. And my thought is that it's not probably terribly unlike what we see with Curculio on oaks, where
Yep.
you've got massive numbers of seeds being produced in a good year. Yes, the beetles are abundant, and there are nuts which are, or acorns in this case, which are infested to the point of destroying them. But there's an awful large number of acorns that escape or have smaller densities, or a few larvae or whatever. Would you agree that that's probably the same case here, where masting, if you will, is such that there's still enough nuts to get around?
Absolutely. Absolutely. That, in my experience, that and from reading literature, that is the most likely scenario, which also would explain why there are some larvae that will overwinter for a second year, rather than emerging with the majority of them the first summer that they have. Sort of a bet hedging strategy, if you will.
You've always observed oviposition on the tree, correct? Or your assumption is that's where it occurs for the most part, right?
Yeah. Yes.
So,
Yes.
so there is the tree, or the nuts, if you will, can escape to some extent by getting to the ground before they've been oviposited upon, correct?
Absolutely. There is some literature. Um, these were experimental grows. I believe it was in Glen, Glendale or Bell, Maryland, back in the 40s, I believe, where they would find it wouldn't be uncommon, given the just vast abundance of nuts at those experimental sites, for the weevils to just be on the burrs that are on the ground.
And you would agree, wouldn't you? Because this is what I've gotten from your work. Lessers are still much more common in the places where the greater, correct?
Absolutely.
Yeah, and so it's still hard to find the greater despite all the energy and time that you put into it. And lessers are relatively common. The greater, for whatever reason, not sure that I understand, maybe you have some thoughts, remain infrequent.
Yeah. For let's see, we found 5 so far, and I would say during that trip in Meadowview alone, I was able to collect about 30 lesser chestnut weevils and only 2 graders. So the lessers are definitely more abundant than the graders are.
We had a question that I wasn't sure whether this was answered by one of you presenters or not, but I see Jim Cartmel is curious about whether you've been able to isolate the genes that are involved in this, I guess, the degradation of sucrose to glucose and fructose, and insert them into anything like yeast to see if they'll digest plant material. I don't know how much you all have done with that type of work, but it's an interesting question. Any additional thoughts on that one?
Yeah, for sure. So the short of it is that we have cloned these genes into insect cell lines, insects SF9 cells. First did that in the years leading up to 2016 when I published the Asian longhorn beetle genome, where we functionally characterized a set of basically lignocellulase degrading genes from that genome, characterizing them functionally, understanding patterns of sub and neo-functionalization in the various copies of those genes present in that genome. That was work done collaboratively with colleagues at the Max Planck Institute for Chemical Ecology in Germany. I'm really happy to say that a former graduate student from that group is now a postdoc in my group, has been for a couple of years, and we've now been doing that work here in-house. And so we're doing two things. On one hand, we're broadly surveying the major lineages of Phytophagus, especially wood-boring Coleoptera, because what we've learned is while they have the same gene families, if you will, have obtained the same gene families, they've undergone these remarkable patterns of sub and neo-functionalization, gene family level, or I should say gene level diversification. And so depending on the various intricacies of the biology of the different species of phytophages that we're working on, we're seeing differences in the genes and what they've evolved to be able to do. So again, yeah, we're doing that here in-house. One of the other fun parts is that I'm interested in different biologies and how that's, dictated the evolution of these genes. For example, some work on Antarctic weevils, which are a real interest of mine for a lot of reasons, including that they function biologically in environments at and near freezing or even a little below freezing. And as ectotherms, these genes, these enzymes are functioning at those temperatures. Their ancestors, we showed a few years ago in a paper in PNAS, are tropical. And so they have ancestors whose genes are functioning at ambient temperatures that are much, much warmer. And you might imagine that there's some pretty neat potential biotechnological application beyond the biology and sort of the theoretical implications. So not to carry on, yeah, we're doing a lot of stuff like that because it's really fun. And we're learning that it's not limited to these genes. There's other genes with adaptive potential that are horizontally transferred along with these in arrays on chromosomes. And we're learning a lot about how that happens as well, and haven't quite published all that yet. They're fun stories, though, that I hope that you'll all, if you will, stay tuned to hear about when they get out in the literature in the next year or so.
We had a question about pecan trees from Linda. She has over 200 pecan trees, and she's not getting production from them because when she finds an adult nut, it has a weevil in it. So this relates to control, and I don't know. I guess I don't know if they're the same weevils that are involved here. Probably not, but maybe there's something you folks could tell her to try to help with that problem.
So, excuse me. They very well could be Curculio cariae, the pecan weevil. Um, I don't know nearly as much about them as I do the chestnut weevils. Um, all I know is from literature, there have been various pesticides and insecticides and chemical treatments applied to these commercially important and economically important trees, whether they be hazels for hazelnuts, pecans for pecans, chestnuts for theirs, or otherwise. And some work better than others, but none of them are really, ooh, this one's really good. And on top of that, I don't have a lot of experience rather any experience with getting rid of insects. I'm usually trying to find them.
I'll just quick comment on that, and there maybe are others in the audience with more experience, but I've done a fair bit of rearing of Cariae, and what I find at least is that they're pretty good at finding the nuts, of course, and gosh, it's really hard without insecticidal treatment. For me, at least, what I've found is similar. Literally every single nut is infested. And circle traps are effective in catching the adults for the reasons mentioned earlier in the chat, that they tend to orient to the trunk and then wander up into the tree. But at the same time, they also seem to circumvent those traps pretty well. It must be by just dispersing directly into the canopy and otherwise. So I don't have anything useful to offer other than that. Darn that evolution, right? It's pretty good at allowing these beetles to find those nuts, and the resolution with which they do it is remarkable. Yeah.
There've been a couple related questions and comments about the prevalence of the larvae in the nuts, and Clark Beebe points out that all you need is one nut to sprout, so that to maintain the population of the chestnut, if each tree produces one offspring during its lifetime. So because they make so many nuts, some of them are, it's the weevils. And I think Michael, you already pointed that out, or maybe it was you, Duane, that when you find trees producing oaks, for example, so much of a mast crop in certain years, it kind of overwhelms the predation impact. But I also, there was a question about, again, back in the day when you had chestnuts roasting on the open fire, and truckloads, railroad cars full of chestnuts being shipped north to sell in for human food. Did they have a problem with massive weevil infestations? I don't know if we really know that, but I think we would have heard about that in some of the historical accounts if it was a major problem. Any thoughts on that?
So there is a paper by Brooks and Cotton from 1929 who used a couple of orchards that the USDA had access to. There was one in Bell, Maryland, and there was one it was either Falls, I think it was Falls Creek Falls, West Virginia. And so these were orchards used for that were there for, I don't remember the purpose if they were initially there pre-blight for commercial use, or if they were established during the blight to try and figure out a way to mitigate the disaster that is the blight. But they report in this technical bulletin that they would get just a vast amount of nuts. And I remember in one, for at least one tree they mentioned, there was upwards of complete infestation for one of them. But that's, in the literature that I've read, that's not very common to have a complete infestation where none of them are viable either for recruitment for new trees or commercial value either.
Yeah, so it would have been sort of an episodic type of thing, maybe.
Yeah.
Dan Lefevre points out that there's some pretty good evidence that at least one person growing pecans gets wonderful control using heavy applications of boron. So I wonder if there's a trace mineral deficiency that that would be remedied by applying boron, if it was boron that was deficient, that could be involved in these cases where you have large infestations. Any thoughts on that?
Yeah, I don't know. Have no idea.
Yeah.
Interesting.
I don't either.
Yeah, it's interesting. I mean, it gets to the point, is a totally healthy organism going to get infected with something? And I think probably the answer is yes, but maybe not so much if there are trace minerals that are involved in the plant's defenses.
Right.
It could be important.
Well, to the comment earlier, which I appreciated, that only one nut has to develop into a mature tree, is the reality too that we have to look across the life cycle of the whole tree, right? That doesn't even have to happen each year.
No, right.
And
Yeah.
so what that suggests, of course, is that there's an awful lot of accommodation of herbivores by these trees, because frankly, any other interpretation doesn't make a lot of sense, I think. You know, and so we see analogs, of course, in other trees in other parts of the world, even the tropics. And it certainly is very interesting to consider in terms of life histories and strategy, if you will. Cool
Yeah.
stuff. Yeah.
Yeah. Here's a question from Hal Brindley. He's wondering, and you probably run into this. And I remember hearing once upon a time somebody said there's two different kinds of entomologists. There's the ones that just want to kill the insects, and there's the other ones that think they're beautiful and interesting. And the question is, do you find it challenging to work with American chestnut people who are mostly interested in killing weevils?
Actually, I found quite the opposite. They were all like, "Yeah, come on, find it. How can we help?" Everyone's just been so helpful, whether it be accommodating me during ramp up to their field season. Because in Meadowview, I went in August, and then the following month they began their harvest, or I think actually a week or so after I left, they began their harvest for 2024.
Good. Well,
So they were, yeah.
American chestnut people are a friendly bunch, I think, generally. So it's been my experience too. Well, this might be a good place to wind it up, unless there's anything else that I don't see any more open questions. And
Mike, can I just share something real quick?
Please do.
I would just offer to everyone on, if you have any thoughts relative or relevant to Michael's work, we'd love to hear about it, whether it's observations. We suspect these weevils have been moved around a bit more than we appreciate or that we know. I don't think they're abundant, Michael. We would agree. We don't think they're abundant anywhere. We think they're probably in other orchards in the region. Would love to know about that. The other is Michael and I both have a shared interest in other specialized herbivores that, whether limited to American chestnut or at least rare and potentially, you know, declining or concerned natives. So if you have any observations like that of other insects, please let him know or let us know. We'd love to hear about it. More than anything, I think, though, I just love the chance that Michael's had to share this work with you. And my hope is he'll be able to continue in some of it beyond all of this. So.
Great. Well, I think this is a great place to stop, and this has been wonderfully interesting, and it won't be long before this joins the crew of other chats that are archived. So thank you both again, and good luck with all your work in the future.
Likewise.
Thank you.
Okay, we're gonna end now. Thanks, everybody. Bye.
Bye-bye.